Okay. Hi, my name is Evan Apollinar, and today I will be discussing herpes or herpes virus, which is a persistent pathogen. And this photo is of herpes. So first off, herpes is a very common infectious disease. Um, it's all around the world, and once you're exposed to it, you're exposed. Um, and you are always going to be exposed to it. Um, but spoiler alert, once you have it, you can't fully get rid of it. And you will always have reoccurring symptoms if you do have it. So historically speaking, the term herpes is a Greek that it's derived from a Greek word meaning to creep, and so you'll understand more once I go through this presentation because it does creep up. And so Hippocrates was the first to describe lesions that could have been caused by herpes simplex virus. And so in the old days, they would have cold sores, um, from kissing or things like that. Um, Taxonomy. So we have first off our order, which is the Herpes virilis, which is then to our family, it's the Herpes viridae. And then we have subfamilies that will go to the alpha, beta, and gamma of the Herpes viridae. And then specifically speaking, we'll be talking about the simplex virus, which we have the HSV1 and the HSV2. Um, these are more strains that are common, like the B virus or the VZV. But for right now, I'll be specifically talking about HSV1 and HSV2. So here's a picture of our herpes simplex virus, and HSV1 and HSV2 are very similar, and so they're going to be um, kind of made the same way. They're going to have the same structure, and so genome is a linear double-stranded DNA virus, which you can see inside the capsid, which is that green thing. And so it's enveloped in it's called the icosahedral capsid, which it has more sides, but this is a 2D picture, obviously. So the 3D version of it kind of looks kind of like a soccer ball. Um, and so the genome will encode for about 80 proteins, in which half of them are used for replication, and the other half would be used for interacting with the host cell. And so you also have these tegument fragments around the capsid, which will be like you'll have these tegumi capsids, um, tegument capsids right here. They're kind of faint, but those are virus encoded proteins. And then you'll also around this you'll have the proteins, the envelope proteins, which will help bound or bind to the cell wall. And then you have the obviously it's a lipid envelope right here. And so again, the important key strains that we'll be talking about are HSV1 and HSV2. You also have the varicella zoster virus. And so the difference between these would be HSV1 is typically referring to oral herpes like cold sores, and HSV2, which are genital herpes, which are transmitted sexually. You have VZV, which are chickenpox and shingles, and then you have an Epstein-Barr virus, which are infectious mononucleosis, and those are typically assisted with cancers. Um, Now I do have pictures, so just warning you, it's not too bad, but there are pictures. So first off, we have a cold sore, which is referring to HSV1. Then you have HSV2, which are genital herpes, and then you'll have chickenpox, which are referring to the VZV. Okay, next we have the transmission. So HSV1 are transmitted through oral contact, like kissing with your saliva. HSV2 are transmitted with sexual contact. VZV will be transmitted through respiratory droplets like coughing, sneezing, of those sorts. Um, okay, so pathogenesis. So this is just a little overview. I'll go into depth throughout the PowerPoint, but just an overview. You'll have the primary infection, which will enter through mucosal surfaces, which will be like your nose or your mouth, or broken skin. Um, and the thing about herpes is that they have a latency period, and so they can choose, or they don't choose, but they can go through 3 steps of latency of a gene expression. And so I'll talk about that more, but they can hide and kind of wait to be expressed. And they also have a reactivation, which can be triggered by stress, immunosuppressive, or hormonal changes, and which then would be spread to cell-cell fusion, and then they also evade our immune system. So next, or first off, you have your entry, which, like I said, it goes to the mucosal surfaces, and then it'll infect mostly your epithelial cells, which will be, the outside layer of your cells. Um, and then the glycoproteins will attach to the host and then fuse within the cell wall and then deliver the genome, which will then go to the nucleus of the cell. And so here's a picture where you see the glycoproteins are connecting and then it's slowly fusing with the cell wall. And so you can see that this capsid is being released into the cell wall or into the cytoplasm, which then goes to the nucleus. So that's the first part of entry. Then you deal with replication, which is, well, it first starts off with gene expression. So there's 3 different types of gene expression. It'll be early, immediate early, or late genes. And so you'll see in this photo that above here, you see that it's going into the cell wall, into the cytoplasm, and then it'll go down, and then the DNA is being inserted into the nucleus, and it's double-stranded DNA, but it's linear, and so then it becomes a circle. It's called circularization, and then it goes through 3 different types of gene expression, which will either be immediate early transcription, latent transcription, or early transcription. Um, and that way, then it goes through the rolling cycle replication, which is where it's actually replicated, and then it'll go out of that replication cycle into where it is. It'll go inside a capsid, and then it's DNA packaged, and then it goes through the process, and then it'll leave the cell ready to infect another cell. And so next, our immune response. So once that happens, or once the first, um, I guess once we're first infected, then we have an innate immunity, which recognizes that the HSV is in there through the PRRS, or the cytoplasmic DNA sensors, um, and so that way the cell will then start signaling the immune system for help. And so we have these things called interferons, which will help us detect for, um, like, we'll stop. So interferons help inhibit the replication of viruses, which is good. We don't want it to replicate. And then you have a natural killer, which will then see the disease and be like, oh, this, you're not supposed to be here, and kill off that. However, herpes does a very good job of hiding itself, um, which I'll go into later. And then you also have adaptive immunity. So the CD4 with the helper T cells have macrophages and cytokines to help um, against the virus. And you also have CD8 plus and cytotoxic T cells that kill the infected cells. But like I said, so herpes goes through viral evasion, which is they'll go through latency and they'll hide in sensory neurons. And so they don't express their proteins, so they're non-detectable. And so if like our CD8 plus is looking for them, they don't know that they're in the cell, and so they won't kill off that cell. And so then you have, so they block antigen presentations. So like when you, it reduces our MHC class 1. And so when they do that, it limits our immune surveillance. And so they don't know that the cell is infected, and so they'll pass by and not realize that it's infected and leave. Um, the interferons that will help prohibit the replication of viruses, they'll also inhibit that, so it won't, they can't do their job of stopping the virus from spreading. And so around the world, HSV1 is in 67% of the population under the age of 50, which is about 3.2 billion people. HSV2 is in 13 of the population, but the age was spanning from, it was like teenagers to late 60s. And so the people at risk are immunocompromised people, such as also pregnant women, and then sexually active people. So anybody that is sexually active should be careful of herpes. Um, so the symptoms for HSV1 are cold sores, fever, blisters. HSV2 would be painful general ulcers, fevers, and then the VZV would be vesicular rashes or intense pain and shingles. And so to diagnose this, we use PCR, which detects the DNA. And then we also have serology, which differentiates between HSV1 and 2 to determine which one you would have. And then you also have clinical signs, like signs that you could see, like a cold sore or, things around your other areas. And so for treatment, we have antivirals, but of course, there's no cure for herpes. So you deal with the herpes, and the symptoms would flare up. And so the antivirals are there to lessen the amount of times you deal with the symptoms and the times, and also how severe the symptoms are. So those are what those are for. And then in order to prevent herpes, one is safe sex, two are antivirals, and then we have a VZV vaccine for shingles and stuff like that. Yes. So research. So vaccines, we have HSV vaccines are under development. We do gene editing for the CRISPR-based approaches, which right now Harvard is doing a study, or in 2019 they're doing a study where they are inserting a gene to see if they could find the part where the genome lines in the for the virus, and seeing if it could cut it off so that it isn't part of our system anymore. And so that's what they're working on with gene editing. And then they're trying to improve the antivirals, um, the ones I showed earlier. And then the public health in general is trying to be or give more awareness to herpes in order for people to limit the amount of people that there are getting infected. And that's it.